We're going to start discussing the nervous tissue cells found in the body. There are two cells, the neurons and the glial cells. The neurons are the typical cells that are seen in the under microscopes. There are these really large ones with three major parts to it. Those three major parts being the dendrites. These are little extensions that stick off of the neuron. The cell body, which is where the nucleus and a majority of the organelles are found, and then the axon, the portion that sends the electrical signals. The neurons make up around 20% of nervous tissue cells, and then the majority of the nervous tissue is going to be composed of neuroglial cells, also known as glial cells. In general, when we talk about glial cells, they're going to support and nourish neurons, and they make up around 80% of the nervous system tissue. Now, since we have these two major different types of cells in the nervous system, we need to talk about terminology and go over a little bit of the definitions between the two. So there's two major divisions, the central nervous system and the peripheral nervous system, and they're both going to contain neurons and glial cells. What we have to remember, though, is that the neurons are going to have three major parts: axons, dendrites, and then the somas, or cell bodies. So when we talk about neurons within the central nervous system, what's going to happen is those neurons are going to be bundled together. And when they become bundled together, we're going to have all of the axons going to be bundled together in certain areas, and all of the cell bodies are going to be bundled together in certain areas. The axons, when they are bundled together, are going to be called tracts in the central nervous system. The tracts will be myelinated, which means that they will appear white or contain the myelin sheath on them, which makes them white matter. So in the central nervous system, bundles of axons are going to be known as tracts. Well, if the axons are bundled together, then the cell bodies are going to need to be bundled together. Bundles of cell bodies in the central nervous system are known as nuclei. Now, the neurons in the central nervous system are really going to have the same type of functionality as the neurons in the peripheral nervous system, but the difference is going to be the names. And these names are helping us determine what area we talk about. If you hear the word tract, then you know that they're referring to somewhere in the brain or spinal cord. If you hear the word nuclei, you know they're referring also somewhere in the brain or spinal cord. The nuclei are going to be bundles of cell bodies, while the tracts are going to be bundles of axons. Well, we're still going to have bundles of axons and cell bodies in the peripheral nervous system, but we're going to call that something else. In the peripheral nervous system, when we have bundles of axons, we refer to those as nerves. So what this picture shows you is it's showing you individual axons, and they're all going to be bundled together. And then all of these axons that get bundled together are going to be packaged into a larger structure that is known as the nerve. Now, the same process is going to happen in the central nervous system. I just don't have any pictures of that. So we can see that we have individual axons here. Each of these dots is an individual axon, and you can see that we're bundling all these axons together. When we bundle all these axons together in the peripheral nervous system, it is known as nerves. In the peripheral nervous system, the nerves can be myelinated or non-myelinated, so they may or may not contain white matter. And then if we're bundling the axons together, we're going to also bundle the cell bodies together. And in the peripheral nervous system, bundles of cell bodies are going to be known as ganglia. So what this picture is showing us is it's showing us individual cell bodies. So here is a nucleus of one cell body. Here's a nucleus of another cell body. Here's a nucleus of another cell body. So we have hundreds of thousands of potential cell bodies all going to be bundled together in one area. When we see this in the peripheral nervous system, again, it is known as a ganglia. So if you hear the term ganglia, you know that you're referring to the peripheral nervous system. If you hear the word nuclei, you're referring to the central nervous system. But both are describing bundles of cell bodies. If you hear the word tracts, you're referring to the central nervous system. If you hear the word nerves, you're referring to the peripheral nervous system. However, they're both referring to bundles of axons. So the terminology helps us distinguish the location in the body. Now, before we can move on and actually understand how the nervous system sends signals and supports the neurons that do send the signal, we need to understand the parts of the neuron. So we're just going to go over the parts of the neuron. Now, this is a specific type of neuron we're looking at that we'll discuss in a minute. However, all parts are going to be the same on different neurons. The appearance just may change depending on the type of neuron we look at. So the first part is going to be the dendrites. The dendrites are cytoplasmic extensions that are going to receive information. So we can see right here we have this arrow going towards the axon. So dendrites' function is to receive information. Next we have the cell body, also known as the soma. So you may hear the term soma instead of cell body. The soma is going to be the area that contains the majority of the organelles. Obviously, the nucleus is in the soma. We will have cytoplasm and mitochondria, Golgi apparatus. But some of the more important organelles are going to be the neurofibrils and the nissl bodies. The neurofibrils will be found throughout the entire neuron, and they're going to help with movement of organelles. They're going to help with division. They're going to help with structure. Then also we have nissl bodies. Nissl bodies are rough ER within neurons. If you can remember what rough ER does, rough ER helps produce proteins. Well, the nissl bodies do the same thing. The nissl bodies will produce proteins that could potentially be packaged as neurotransmitters, which means they're going to be chemical signals eventually. Now, coming off of the cell body, you are going to see a cone-shaped area. We call this the axon hillock. So this cone-shaped area is the axon hillock, and off of the hillock we have our initial segment of the axon. So the axon hillock's the cone-shaped area, and then the initial segment's just the beginning of the axon. The axon hillock's important because the axon hillock is going to contain what we call the trigger zone. The trigger zone is where new electrical signals or new action potentials will begin. So the axon hillock contains the trigger zone, and this is where new action potentials will begin. So the axon hillock leads to the initial segment, which is going to be the beginning of the axon. The axon's job is to send electrical signals. Now, the axon can be long or short depending on the neuron in the body. And at the end of the axon, what you're going to see is these little extensions, and these are known as the axon terminals. Terminal means end, so these are the extensions at the end of the axon. But you'll notice that those extensions end in a swelling. These are known as the synaptic end bulbs. It's within the end bulbs that we are going to see neurotransmitters packaged in vesicles, which we've already discussed in the previous chapter, chapter 10. Now you'll notice in this particular picture, this axon has material on it. This is where we're going to see the nerve impulse jump over, and it's jumping over areas called the Schwann cell. Now the Schwann cell is going to be a special cell that helps produce the myelin sheath, which is a lipid protein covering. Now, since it's a cell, it's going to have a nucleus, as you can see right here, and it's going to have its own plasma membrane, which we call the neurolemma. So if you remember when we talked about the muscle cells, they had sarcolemmas. Well, Schwann cells are going to have neurolemmas. So we can see that we have this white area. This is the lipid protein covering that makes up the myelin sheath. And then this outer layer, just this outer layer right here, is going to be the neurolemma, which is actually the cell membrane of the Schwann cell. What the Schwann cell does is when it myelinates, it basically helps insulate the neuron, and it's going to help the nerve impulses hop from this gap to this gap. These gaps are known as the node of Ranvier, and we can have hundreds of gaps or we can have just a few gaps. It depends on the length of the axon of the neuron. Now, because neurons are going to be found in different areas of the body and because they're going to look different, we're going to have different structural and functional classifications. If you remember, we talked about function already. We can have sensory neurons. They're going to take information from the peripheral to the central nervous system. They can also take information to the brain. We're going to have interneurons. They're going to be the integrative or analyzing neurons. Those are only going to be found within the brain and spinal cord, so the central nervous system. And then we can have motor neurons. These are going to take information away from the central nervous system, or they can take information away from the brain. So that's function. But then we have structural depending on their looks. So when we talk about structural, the first thing we're going to talk about is how many projections do we have coming off of the soma. So if you remember, let's go back to this picture real quick. Here's our soma. And then we have different projections coming off the soma. Here we have lots of dendrites, and then we have our axon. So when we talk about structural, we talk about how many projections come off of the soma. So our first one is a multipolar. Notice that we have lots of dendrites coming off. And then we have our axon. So would you say this is one, two, or lots of projections coming off the soma? If you said lots, then you're correct. Anytime you have lots of projections off the soma, it's going to be referred to as a multipolar neuron. You'll notice that we still have all the same areas. We have the trigger zone within the hillock, axon, myelin sheath can be present. We have our axon terminals or synaptic end bulbs. Multipolar neurons are going to be motor neurons. So anytime you hear someone talk about multipolar, they're referring to either motor neurons, which is the majority, or they could be referring to interneurons. Okay, our next type is going to be a bipolar. When you hear the word bi, what do you think of? If you said two, you're correct. Okay, here we have one major dendrite that splits and branches, and then we have our axon. So we have one projection here and one projection here. So that's two, which tells us it's a bipolar neuron. Notice we still have the same parts. We have our cell body, our dendrites, our trigger zone, which is the hillock, our axon, we have our terminal, and our end bulbs. Bipolar neurons are unique because they're only found in special senses, such as your retina for vision, your olfactory neurons, your gustatory, your taste neurons, and then your auditory neurons. So we only find bipolar neurons in special senses: eyes, nose, ears, taste. Our next will be uni. Uni means one. So if you look here, we have one projection off of the soma, and then it splits. One end goes to the dendrites. The other end's going to be the axon. We still have the same parts, though. We have our trigger zone where the hillock will be. This is what turns into the initial segment of the axon. We have our axon terminals and our end bulbs. Unipolar neurons are going to be your sensory neurons. Basically everything except for special senses. Now, what's unique is that if you think about some of these sensory neurons that we've talked about, or the sensory receptors, we've talked about Merkel's, Meissner's, Pacinian, we've talked about pain receptors. Well, all of those that we talked about are considered sensory neurons, unipolar. So here is the Meissner, detects touch. Basically, that's your dendrites. Your dendrites will lead directly to your axon. Here's the Merkels. They also detect touch. These are the receptors on the dendrites. They lead directly to the axon. Pacinian, deep touch, deep pressure. Here's basically the dendrites leads to the axon. And then nociceptors, these are for pain. These are the dendrites. They lead to the axon. Why would it be important to have these as unipolar instead of multi or bi? Well, what we're skipping is we're basically going through the integration area of the soma. So instead of having to go through 3 steps, the dendrites, the soma, and then the axon, all we have to do is basically go directly from dendrites to axon. We get to skip step 2. So this makes the sensory receptor much faster. So think about if you put your finger on a hot stove, you want to have a very quick reflex. Well, the faster the sensory receptors can communicate and send a signal, the faster your reaction will be. Now, our next cells are going to be the glial cells, neuroglia cells. There are going to be a total of 6 that we're going to go over. 4 of them will be in the central nervous system. 2 of them will be in the peripheral nervous system. Now, this picture that we're looking at right here is of the central nervous system. Remember, the majority of the function of glial cells is to support and nourish the neurons.